Good morning, everybody. I'm Christine Saletti, and I'm a facilitator for the ThyCa in South Florida. And I am here to introduce Thomas Giordano. I want to remind everybody to thank all the doctors that come and speak to us. We're a total nonprofit organization, so that means they're on their own dime, spending their own money to come here and their own time. So we sincerely appreciate the knowledge we can gain from all these doctors that come and volunteer to speak to us. So let's give them a round of applause. Dr. Giordano co-authored a paper. He's a doctor and a professor at the University of Michigan, and he co-authored a paper about the biomarkers that are found in the pathology of thyroid cancer. So he's here to explain it to us in the most layman terms possible. But
I will do my best to try. So it's a great pleasure to be here, and thank you for coming. I know the title's not the most exciting or inducive. Let's see, how does this work here? So I'm just supposed to say this. I doubt there's any reporters here, but we're publishing this paper next week in Cell, which is sort of a high-profile, biology journal, and they embargo the press. So if there's any reporters here, you can certainly write about it, but you can't have your paper come out until the press embargo is up next Thursday. So Gary just wanted me to say that, because I would be in a lot of trouble if there was something. So just a disclaimer, I'm a pathologist. I'm pretty much behind the scenes. I spend my days talking to people that work in my research lab. I talk to surgeons, endocrinologists, all kinds of other doctors, and my pathology colleagues. But I do talk to patients when they reach out to me. I think a smart patient finds their pathologist, because as I'll go through this, we sort of seal your fate in many ways. And so making sure that your pathology is accurate and done by someone with experience is kind of important. So I'm here all day today, and I'll be glad to chat if anyone has any questions. So here's the outline of my talk. I'm going to introduce a few general concepts about the cancer genome, genetics, etc. Talk about the role and the current state of molecular testing for thyroid nodules, and then introduce the Cancer Genome Atlas project, and then sort of tell the thyroid story. This has consumed my life for the last year. We started writing this paper last October, and we're writing it all year, submitted it in May, and it's coming out next week, as I said. It's a big and complicated story, so I'm going to try to hit the highlights, and again, I'll do my best to make it as understandable as possible. There's only so much, I can't change the data, so it's a complex story. So what about some of the key concepts about genetics and genomics? So this is for people who are interested, this is a great review article. It's a little dated now, 2009, but the concepts are certainly there. And so basically it's talking about that all cancers arise as a result of somatic, referring to all the cells in our body as opposed to the germ cells. So all the cells, our skin cells, all the cells, changes acquired in our DNA. And that's what we've come to realize. That cancer is basically an acquired genetic disease. And but along the course, we get two different kinds of mutations. We get mutations that are causative, that help those cancer cells grow better and more better than the non-cancerous cells. And those mutations we call driver, because they're driving the cancer. But in the process of just having your cells divide, and we have 3 billion bases and each cell has 3 billion bases in its genome. When you go from father cell to two daughter cells, and you replicate that DNA, that's an inherently mutagenic process. You get mutations, but a lot of those mutations don't do anything. They're along for the ride, and as a consequence, they call those passenger mutations. And so that's one of the challenges of this field, is when you do the sequence of someone's tumor, you find all kinds of things. And it's hard to know what's the drivers and what are along for the ride, what are the passengers. So those are key points that we sort of have to understand. And it sort of this is from that same picture, and this really drives from the point of sort of big picture, why does cancer exist? And it just shows sort of the lineage of mutations through life, from fertilized egg, gestation, infancy, childhood, adulthood. And then they divide up the causes of changes in our DNA into intrinsic mutational processes. Just living, just copying our cells every day causes mutations. And it starts right away at the beginning, from fertilized egg into gestation. Mistakes happen throughout our life. Environmental lifetime exposures. So, having charbroiled steaks all the time, there's carcinogens in there that cause mutations. So environmental exposures. And then some people have a mutator phenotype. I don't know if you've heard about syndromes like Lynch syndrome, where you have a defect in your DNA repair mechanism. And so not so much that you make more mutations, is that you repair them less well. So the point is you make more mutations, and so a mutator phenotype. And then patients that actually get like cytotoxic chemotherapy, that's known to be mutagenic. And so you can see the chemotherapy-resistant cancer cell has accumulated a lot of mutations. And one of the rules for the TCGA project was no chemotherapy. The tissue must have been collected before any chemotherapy was given, because we wanted to avoid finding all these mutations that were related to the administration of chemotherapy. Okay? So this is an amazing slide. It sort of puts things in perspective. And it sort of ties in with this editorial, which was in the Times, less than a year ago. And I thought it was very interesting why everyone seems to have cancer. And right here, mutations are the engines of evolution. Without them, we would never have evolved. The trade-off is that every so often, a certain combination will give an individual cell too much power. It begins to evolve independently of the rest of the body. Like a new species thriving in an ecosystem, it grows into a cancerous tumor. For that, there can be no easy fix. So in some hands, this article is sort of saying, this notion where we're going to never have cancers develop is almost a little naive, right? And basically says if we live to 150, people are eventually going to get cancers because of this intrinsic process of copying our DNA all the time. Now, I like to read the comments on the New York Times, and a lot of people were upset with this editorial because a lot of people wrote back and said, if I just live a good lifestyle, you'll be fine, don't worry about it. I really like this one, though, from Jane from Pasadena. She said, great op piece. She sort of got the point. Now I can worry about if my DNA is replicating itself properly, sans mutations for the remainder of my life. I'm already stressed enough, right? So you never know how your work is going to be perceived. Poor Jane, she's stressed out. So that's a little bit about the genome stuff, and I really wanted to spend some time with that because I think it's important. So what do I do? I'm a diagnostic surgical anatomic pathologist, right? So just yesterday, we did a frozen patient with an ovarian tumor, and looked like a neuroendocrine tumor. We said, could this be a carcinoid? And the surgeon, I'm on the phone with the surgeon. She said, actually, there's a small bowel tumor. And I said, ding, 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 ding. This is probably a carcinoid of the small bowel that's metastatic to the ovary. So we talked to the surgeon. So we're in the trenches helping surgeons figure out, what to do in the OR and writing final pathology reports. But basically, this is the process that we go through every time we pick up a patient's class slide. What is the disease process? Is it a neoplasm? And if it is it cancer? If it is cancer, what kind? Is it an epithelial tumor that we call carcinoma? Is it a mesenchymal tumor that we call sarcoma, melanoma? Is it some sort of leukemia or lymphoma? And then if it is, what type? If it's a thyroid carcinoma, is it papillary? Is it follicular? And then beyond that, we were always expected to sort of do all the things that say, how bad is it? What is the stage? Is it confined to the thyroid? Is it grown into the lymph nodes? Are the margins free, et cetera? So these are all the things that they're the algorithm that we go through as pathologists to write a pathology report. And ultimately, these answers to these questions dictate how people get treated, right? And really, no one gets treated without a finalized pathology report. So the oncologist will call sometimes if the path report's taking time. So they're like, we need to start this patient on chemotherapy, tomorrow, but we're not going to do it until you finalize that path report. So, because the last thing we want to do is give chemotherapy A, and then the pathologist say, by the way, we changed the tumor from this to that, and then, right? So, until this is verified, nothing really happens. So that brings us to thyroid and sort of molecular pathology of thyroid nodules. And the first question is, why bother, right? Pathologists we do pretty well with the microscope. Why bother? And one of the things that allows us to do this whole molecular analysis of thyroid nodules is the fact that there's a strong correlation between the genotype, and genotype means is a sum of just the mutations found in a sample, between the mutations and its morphology or its architecture, basically what I see through the microscope. So there's a correlation between what I see through the microscope, and so to the point is, now that you've done enough of this, I can actually look through the microscope at a tissue section and say, oh, this tumor probably has a BRAF mutation. So that correlation is so strong that I can say that with a lot of comfort. I don't write that in my path report, but sometimes I write, this tall cell type, most likely has, we can get away with that. And so this correlation allows us to exploit this relationship in cases where morphology is lacking. And in particular, it's lacking when you put just a small little needle in someone's nodule and suck out a few cells. That's known fine needle aspiration. And so limited samples. And I'm sure many of you had fine needle aspirations, and sometimes the path report sounds like mumbo jumbo and you're not really sure what it means. And so that's just a reflection of two things: limited sample, and the fact that sometimes the evaluation of thyroid tumors depends on the assessment of invasion at the periphery. And when you put a needle in the middle, you're not assessing invasion at the periphery. So it's just an inadequate assessment. Sometimes pathologists disagree, right? So there are cases that cross my desk that are called thyroid cancer, and I sometimes change them, and vice versa. So, and this is well documented in the literature by getting 10 thyroid experts to review, 100 cases blinded, and the diagnoses are not in very high agreement, depending on the case, depending on the type of tumor. I don't want to give you the impression that we're useless, but there are certain types that cause a lot of differences in opinion. Diagnostic interpretation. And thyroid is one of those fields that would benefit from something more objective, and hence the need for molecular pathology. So a little time capsule. Driver mutations in thyroid, we had a small list. We have RET gene, the BRAF gene, NTRK1, RAS, this gene fusion called PPAR gamma, and then this is a gene called beta-catenin, and P53, which is sort of the gatekeeper of the cancer genome. P53 mutations are associated with much more aggressive tumors because P53 basically, if a cell gets damaged, when it's healthy, it tells that cell to die because it's been damaged too much. When P53 is mutated, that cell lives, and that damage persists. And so when you have a tumor that has a P53 mutation, bad cells that shouldn't be allowed to live persist. But the thing is, it occurs late, usually in undifferentiated or anaplastic thyroid cancer, and it's very rare in well-differentiated thyroid cancers. We've learned a lot since 2006. We now have a longer list of mutations. It's grown a little bit. We're getting there. And it's helpful to think about where these mutations are in terms of the proteins and what the proteins do. And in fact, many of the mutations are in genes that encode for proteins in this signaling pathway, the mitogen-activated protein kinase pathway, which has a growth factor, which in this case is RET. It has RAS, and then it has BRAF. So they all sort of line up in the same pathway. So this signaling pathway is incredibly important for most thyroid cancers. And so we were able to draw little models like this. We have a simple model. Excuse me. It starts with a follicular cell. And if you pick up a BRAF mutation from some of those intrinsic processes that we were talking about, or whatever lifestyle, and you're likely to develop a papillary thyroid cancer. If you pick up a RAS or a PTEN or PIK3CA mutation, it'll be a little more in what we call the PI3 kinase AKT pathway, and it'll go down more of a follicular adenoma, follicular carcinoma pathway. And then if you accumulate more mutations, and luckily this happens rarely, you go on to develop more aggressive forms of thyroid cancer that were called poorly differentiated and anaplastic. And the key point is, throughout this whole process, we're going from a highly differentiated state, a normal follicular cell, to basically an undifferentiated state. So there's progressive loss of differentiation through this model. And so the molecular testing field falls out largely in two big categories. There's two approaches to do this testing where you could do testing on someone's thyroid nodule. There's a genotyping method where you're looking directly for those oncogenic driver mutations that are associated with different types of thyroid cancer. So BRAF and RAS and RAS. And if you find those, that tells you something. And ThyroSeq is the assay that Yuri Nikiforov has developed at the University of Pittsburgh using a lot of this genotyping data. Conversely, you can do a gene expression profile, and we published back in 2005 and showed that these different mutations lead to different gene expression profiles of different genes, but nonetheless they were distinct enough that you could associate them and into different classes. And so you could develop a gene expression profiles that are associated with either benign or malignant disease. And there's the Affirma test that's out in the lobby that was sort of based on that. So these are two sort of complementary ways to do the same thing of basically saying, you have a thyroid nodule, what's the risk that it's cancer? What's the risk? What's the likelihood that it's not cancer? Okay? And so what are some of the pros and cons of these two things? The genotyping, it's really relatively easy to add a new gene, right? So you may have 20 genes that you're looking for, and then, like in our paper, we discovered a new cancer gene for papillary cancer. Yuri's already added that to his profile. It's very easy to do. Just add a new gene because of the technology is such that you can just add a new gene. So it's easily expanded to include new alterations as they are discovered. Yet we still don't know we don't know everything. The TCGA paper that I'm going to show you gets us there, but as I will explain, we only did papillary thyroid cancer. We wanted to do more, but the NCI said, well, you have to pick and choose, and it's a long story. So the genetic complexity of thyroid cancer is expanding as all these genomic studies are being done, and it's hard to develop a test that works for every known alteration, especially as we find alterations that may be present in one patient, right? And so you're going to develop a test for the mutation that's present in one patient out of 500. That may not be too feasible. And so there's trade-offs related to cost and practicality and how it relates to how frequent these mutations really are. And so here's Yuri, and here's one of his landmark papers where they looked at over 1,000 fine needle aspiration sessions and showed that molecular testing really did impact the diagnosis of these nodules. And this field is mature enough now that Yuri is writing review articles. Here's a figure from a review article where you start with a nodule, you do cytology, you get some sort of indeterminate result, you do molecular testing for, in this case, just a handful of mutations, BRAF, RAS, RET, PAX8, PPAR gamma, and they're either positive or negative in the different categories, and you can see the risk of cancer changes substantially. So if you're a follicular lesion of unclear significance, uncertain significance, and you have a mutation, the risk of cancer is 88% versus only 6%. And so you can modulate how you treat people based on these results and basically improve the diagnostic power of cytology. Pretty straightforward, but very, very effective. And now what Yuri's doing is taking, as the genomic universe of PTC and other cancers are expanding, he's making this gene list longer. So these numbers are already outdated because his assay is better because it's looking for more mutations now. Medicine moves forward. Here's the gene expression one. Dr. Haugen spoke this morning, published in the New England Journal. This was a gene expression assay that measured the expression of 142 genes to either diagnose or call aspirated nodules either benign or suspicious. They kept it really simple. It's benign, you don't need surgery, or it's suspicious and you may need surgery. And why did they do that? Because this is a little fuzzier, and to get too granular, they really didn't have the power to become too granular. So by keeping it into benign or suspicious, they were able to get a good result. One of the issues that I've heard, though, from other institutions that are doing a lot of this is that patients that might actually be watched, when they come back with a suspicious result, it sort of tips the balance and forces those people to go to surgery, right? And the ultimate goal of all these assays is to more precisely determine who really needs to go to surgery. Because truth is, we do about 50,000 thyroidectomies in the States every year, and most of those are still for benign nodules. And so that's a big cost on our system. If we could effectively reduce that number, it'd be better for patients, better for the whole system. And so they designed this way so it would have a high negative predictive value. What does that mean? You basically don't want to do this test and then miss a cancer, right? So you want to really, if you say benign, you better be sure it's benign, because now you're not operating on someone, and if it turns out to be a cancer and and there's a big delay, that's a potential problem. So they rigged their assay to have a high negative predictive value, so that only in a few times did they have a false result. So molecular testing, I think, of aspirates is improving the diagnosis of thyroid cancer, yet the genotypic testing is limited by incomplete knowledge of the mutations that occur in thyroid cancer. So, and just a little bit about prognostication. I know Dr. McIver talked a little bit about this morning, but it's pretty traditional, conventional clinical pathologic evaluation: patient age, gender, tumor size, whether it's left the thyroid, lymph nodes, distant metastases, and whether other advanced stage. Histologic type, we recognize that the tall cell tends to be a little more aggressive than conventional PTC, and the follicular variant is probably relatively a little more indolent. And then sometimes cancer is not a stable disease. It evolves, accumulates more mutations. Occasionally, PTC can evolve into more aggressive forms, poorly differentiated anaplastic carcinoma, and those are clearly poor prognostic signs. And then so risk stratification, Dr. McIver talked about this morning. They take all these things into consideration and come up with, are you a low risk, high risk, et cetera. And these things determine lots. They determine the extent of initial treatment, whether you get a completion thyroidectomy, whether you get radioactive iodine, how carefully you're followed up in the clinic. All these things come into play with sort of risk stratification. But occasionally we get it wrong. Here's a quote from Ming Zhao Zing at Johns Hopkins, who wrote a review article from 2007, and he said the reliability of this clinical pathological criteria-based approach, however, can be uncertain, particularly in patients with conventionally low clinical pathologic stages. So basically, it's fessing up that we could do a little better at risk assessment. And so, can we do better, and if so, how? So the field is, for the last many years since BRAF was discovered, there's probably 100 papers on what's the significance of BRAF for papillary thyroid cancer. And so you can genotype, here's V600E, but there's a few other mutations, and again, all the molecular biology that goes along with this. However, it's not all that powerful. So this is risk of recurrence, and you can see patients that have a BRAF mutation have a slightly higher risk of recurrence, both in a Caucasian and in the Korean populations. And the consensus view is that BRAF does provide some independent predictive value, but it's not that strong. And if you think about it, it's just too frequent to be all that strong, right? So about 50 or 60% of patients have, with PTC, have BRAF mutations. So how could that really be identifying a high-risk group all by itself? It's just too common. So clearly, there's more things at play. Biology is always complicated. So if we start here with BRAF V600E, you can see, and I'm not going to go through all this, but you can see there's a lot of additional alterations, both accumulation of mutations, gene expression changes, that all end up resulting in loss of radioactive iodine avidity, treatment failure, and progressive disease. And so how can we capture this? We clearly need to do a better way of capturing and assessing the underlying genetic and epigenetic complexity in thyroid cancer. And so this is the vision. This is the vision of personalized medicine, where we're not going to toss out clinical, pathological, and histologic assessment, but we're going to add in sort of a genotyping and even a genomic assessment and put them all together and somehow arrive at an improved classification and prognostication of thyroid tumors. So this is the big picture. And so for anyone who's paying attention, the genome revolution is transforming every field of biology and medicine, from agriculture, microbiology. It's just, it's incredible what they can do. They can use this to identify viral pathogens. They can sequence like crazy. I mean, the whole field of medicine is just changing. It's right there before our very eyes. And so the Cancer Genome Atlas, so now I'm going to pivot to what I've been working on for the last 4 years. The Cancer Genome Atlas is this study that was started at the NCI, and here's their mission statement. It's a comprehensive, coordinated effort to accelerate the understanding of the molecular basis of cancer through the application of genome analysis technology, largely genome-wide DNA sequencing, but RNA sequencing too. Lots of sequencing. It's a joint effort between our National Cancer Institute and the National Human Genome Research Institute. It started in 2006 as a pilot and was expanded in 2009. And basically, part of it was, could we just do this as a big team? Infrastructure for team science, develop a pipeline that would be scalable, demonstrate the feasibility of doing this and sort of identifying the molecular parts of each cancer type, and then make it all available. That's the most important thing. Make it available for the research community, make the data freely available so that you can generate a hypothesis from this data and go off and do a new study. And so here's the timeline. There was an initial pilot report in 2005. The pilots got started in 2006. They started with glioblastoma. They then did ovary. And then you might remember the world almost fell apart. And we had a stimulus package. And Arlen Specter from Pennsylvania personally saw to it that $10 billion flowed to the NIH. And $375 million of that $10 billion flowed right into the TCGA. So I would probably not be standing here if it weren't for the stimulus package, because I don't think thyroid cancer would have percolated up high enough to actually have gotten done. So, whether you like this or the stimulus or not, but there is a reality here of unintended consequence of the world economy falling apart. Some money did flow for science and medicine. Enough with the editorial. And so now the money's winding down, and we've done, they've hit 10,000 cases of all different tumor types. Here's what the pipeline looks like. It starts with frozen tumors. So thanks to any patients who might have contributed samples. There's this very rigorous pathology review, DNA and RNA extraction, and quality control. It gets sent for sequencing and all these other type of genomic analyses, copy number changes, epigenetics. All the data gets stored centrally and scrubbed and cleaned and high quality. And then there's a genome data analysis center together with a whole team of people called the Analysis Working Group that eventually writes a paper. And this, as I alluded to, is not a quick process. This is a prospective, so you can use retrospectively collected tumors, but they have to be identified and all the forms need to be filled out. And so that can take almost 2 years. The analysis then takes maybe another year of generating all this data, and then I can attest to the fact that it took me a good year to write the paper. Started writing in earnest in October of 2013. So it really did take us 4 years to sort of, the first emails, which I still have, started in January of 2010. So it's been a long ride. And so their view is to not leave any scientific platform behind. So they wanted to do most forms of cancer. They wanted to do almost the best that science had to offer, from all this high-tech methylation, microRNAs, DNA sequencing, gene expression, but also on a good foundation of traditional pathology and clinical medicine. And to do that took quite an investment in infrastructure. So they set up a biospecimen core resource, which is actually there were two of them, one at TGen in Arizona, one at Nationwide Children's in Columbus, where all the samples went, got all treated standardly. They have genome characterization centers, sequencing centers, 7 genome data analysis centers, a coordinating center. So this is big team science, and that's why they spent $375 million. There's a big network that's mostly coastal, but there's Ohio State and WashU, and it's even international because the British Columbia Cancer Agency is in there. And so the nice thing about this is I have friends at all these places now that we get emails from every day, and it's really, I think it really has succeeded in uniting the cancer genome community throughout, because now that TCGA is winding down, we're all saying, okay, what's next? And there are projects. The NCI just announced a program called the NCI Exceptional Responders for people that have responded exceptionally, and they now want to study their genome and try to figure out why that might be. So it's sad, but endocrine neoplasia was almost left out of the whole thing, because if you look at big statistics, you can see melanoma, sarcomas, head and neck squame, thoracic, all these cancers are very lethal with tens of thousands of people dying every year. And thyroid, while it's, it's bad cancer, but, relatively not as bad. And so thyroid, I think, was a push. And I don't know who championed it, but they got it through. Once I became the chair of thyroid, I also am interested in adrenocortical, and we convinced the TCGA, once they took the money for one large project and we're going to do some rare cancers, we jumped up and down and made so much noise that they finally let us do adrenocortical. So we're writing that paper now. And then I don't know who the champion, but there's another adrenal tumor called pheochromocytoma and paraganglioma. So we actually have 3 endocrine projects that made it through the analysis. So, and this is really big data. People talk about big data. Notice breast found the money to do almost 1,200 tumors. But thyroid is here. It's now red because it is, it should be red because it is, this is an old slide. It is impressed. And so let's talk about the big picture. Now that you have all this data from all the tumors, let's talk about the big picture. Now remember, we did papillary thyroid cancer exclusively. And so PTC is here, and this is the mutation density. And you can see, and this is a log scale, so you can see like the melanoma and the smoking-related cancers have the highest mutation density. Makes perfect sense, right? And thyroid cancer PTC is relatively indolent, and it makes sense that it has a lower mutation density. So when a pathologist looks at this slide, it makes perfect sense because it correlates with what we see through the microscope and what we know about the biology. The other thing you can do is you can start to sniff out what are some of the causes. So this is a really complicated slide that defines 6 signatures based on the types of mutations, the specific, T's and G's, and those are related to certain mutagens. And you can actually see that there is a strong lung signature that they call signature 2 that is related to smoking. And embedded in this gamush is adrenal cancer, and you can see that there are some adrenal cancers that are down in the smoking pie. And we're speculating that adrenal cancer, some, may be related to smoking. There was some data in the literature that suggested that, but it wasn't easy to prove, and so this is just another piece in the puzzle. Thyroid is not very specific. It sort of is just scattered about. So there's no clear, mutational signature of PTC. And so the field has been expecting this for a long time, knowing that molecular pathology would improve our assessment of thyroid tumors, diagnosis, classification, and prognosis by building on established determinants like histology and staging, but adding in mutations and gene expression and protein expression and modification, putting this all together to arrive at a better state, a more informed state. And so here we were, a little personal biography. Here we were back in Cambridge. So I was the co-chair for the disease side. The way they do this, they also give a co-chair for the GDAC, the Genome Data Analysis Center, which was this fellow here, Gadi Getz, who's trained as a traditional physicist, got his Ph.D. in physics from Israel, but then discovered biology, and he's the head of cancer computational biology at the Broad Institute, which is in Cambridge, which is a joint effort between Harvard and MIT. And Eli Broad and his wife gave hundreds of millions of dollars to establish this. And so these guys are the real deal. And Gadi is phenomenally smart. But you might notice a few people. There's Yuri Nikiforov and Jim Fagan and Memorial, Chris Umbrick, who's at Hopkins, and Ian Gainley, who's a surgeon at Sloan Kettering. But most of these other people, David McFadden, the young endocrinologist at Harvard, most of these people are mostly computational biologists who don't know anything about thyroid cancer, but they learned. And so that was the whole point, was to bring together a group of computational biologists, a large group of computational biologists, and a small group of disease experts, and work together through the course of phone calls and emails to derive a result. And so back to our model of simple thyroid tumors. So the TCGA said, we don't really like projects that have a real mixture of tumors. Because we actually proposed, let us do this whole thing. Let's do PTC, follicular, poorly different. And they said, no. That'll be a mess. Pick one. And when you have to pick one, 85% of thyroid cancers are papillary thyroid cancer. So by definition, we picked PTC. There was no other choice to get 500 because that was our goal. And then from the histology, they really fall out into three types: the true papillary tumors, a follicular pattern tumor, and this tall cell type. So we tried to keep the pathology, even though there's other rare variants, we tried to keep the pathology pretty simple. And again, there's a strong correlation between genotype and phenotype in here. So I could tell you that this tumor, typically has a RET rearrangement or a BRAF mutation. These tend to have RAS, and these almost always have BRAF. And so here's the data. We generated a fair amount of data across all platforms, 496 samples, sequencing of 402. Some had all 496. And then we actually did 49 whole genomes, where we sequenced the whole genome of the tumor. So incredible amount of data was generated. And back to our mutation density plot, where thyroid is relatively low with 0.41 non-silent. So non-silent is a mutation that may be a driver. It doesn't say it's a driver, but it's likely to result in a protein change. But not all protein changes are deleterious, right? So it gets a little complicated. So this just shows mutation density, and basically shows that some tumors, some minority tumors, have much higher mutation rates than others. But it's a whole range, and it's not due to any kind of technical differences. And so there was a question this morning about age, and Dr. McIver was speaking about why 45, why is there staging of PTC change. We showed that, in general, there's a correlation between age and mutation density, and it's a pretty linear correlation. So nothing magic happens at 45, right? And so we argued that, we did two things. We wanted to say, is this associated with any type of risk? And so to do that, we developed the ATA risk profiles for all of our patients, and we also developed their MESA scores. We got all that data, okay? And then we asked, is there a correlation between mutation density and risk and mutation density and MESAs? And there is. Here's MESAs, but the problem with this is that the A in MESAs is age. So there's a circular, so here's where Gaddy comes in, the brilliant mathematician, says, no, we have to regress out the effect of age from MESA scores and see if this relationship holds up. And so notice the line's going up, you take out age, and the line now turns flat. So when you regress it out, the whole relationship is due mostly to age. So it's a circular argument. But we were able to show that high-risk tumors did have a higher mutation density. So we were able to make a statement that more mutations generally leads to more aggressive cancers. Yes?
I just was curious, are other cancers associated with age also? Is it only thyroid cancer?
No. So it's a great question. We debated this back and forth because I spent a lot of time looking at these cases and became convinced that there was something to this story. But I had to push really hard against the hardcore statisticians. Eventually I won. But and the argument was that there are certain kinds of cancers, like these Lynch cancer patients, colon and endometrial, they have a much higher mutation density, but they don't necessarily do worse. And so Gaddy's argument was, no, but I can tell you, I shouldn't scoop anybody, but there's another genome study that's still in process where they have longer-term follow-up, and basically more mutations in thyroid is generally associated with more aggressive disease. But I think every cancer might be a little different. But back to that main plot, you can see the ones at the end, the melanomas, the esophagus, the lung cancer, those are really bad cancers, and they have a higher mutation density. So I think the relationship generally holds.
Because we were once at a seminar, because there's all kinds of learning that takes place at this conference.
Right.
And this doctor thought that perhaps a person's immune system is not as strong as they age, which causes the more serious cancers because their immune system isn't strong enough. But in that theory, why wouldn't that happen with all cancers?
Yeah, so
Because when you do the prognosis on most cancers,
yeah.
they don't add up age like they do with thyroid cancer.
Right.
Oh, you're over 45, well, you're doomed.
Right.
No,
So our conclusion. So we sniffed out. We had a few tumors that were exceptionally high, and they certainly were more aggressive forms that I could recognize. So the tall cell variant had higher mutation density, statistically significant. And so we made a statement here that accumulation of mutations is associated with more aggressive forms. And this is a statement from the paper that the linear relationship between mutation density and age raises the consideration that age should be considered a continuous variable in risk stratification as recommended by others, instead of applying the arbitrary threshold of 45 years that is used in many staging systems. So someone just made it up. I don't know enough about the history of where they pulled out 45, but...
So they're going to take that out of the prognosis calculation?
Well, so medicine moves a little slowly. This paper will get published, and people will consider it, and it'll be discussed, I'm sure, in the next ATA or staging considerations. But we made our view pretty clear, that we thought that nothing happened at 45, and it was more of a steady accumulation. So this is part of our landscape view, our first figure with some clinical parameters and the mutations. And you can see we have BRAF in almost 60%, and then we have RAS mutations. And notice they're mutually exclusive. We also discovered this gene EIF1AX, and it's right here in a small set, 6 cases. And notice it's mutually exclusive. And what does that mean? It means that there's no advantage for a cell to have more than one of these mutations, right? They're all sort of in the same pathway. So once you have a BRAF mutation, there's no biological advantage of having a RAS mutation. And the fact that EIF1AX is now mutually exclusive is shouting to us this is a new driver gene. On top of that, Gaddy's group was able to show statistically that it's likely to be a new driver gene. So we found significantly mutated genes, the BRAF, the RAS genes, EIF1AX, CHEK2, and PPM1D. So we found some new genes associated with thyroid cancer. Here's EIF1AX, and it's turning up in other cancers, uveal melanoma, and some other cancers shown here. And our mutations here lining up with mutations from other tumors, et cetera. So we made a strong statement that we think we found a new cancer gene. Yuri's already plugged this into ThyroSeq2, and he's already finding it. Question?
How do you measure mutation density?
So it's just basically you know how big the genome is, and you know how many mutations you figure. So it's just basically number of mutations over number of megabases sequenced.
So if it's and I'm a little basic here, if you have a BRAF mutation,
That's one mutation.
So,
V600E, that counts as an N of 1.
so how would you, are you saying there, I'm having a tough time making the leap from, you have got one mutation, how is that measured in terms of density?
So we don't have one mutation. In those tumors that BRAF V600E, they might have 200 mutations, right? Because the genome is 3 billion bases long, and we've sequenced the exome of that whole genome. So we've looked at that almost all the genes, and we've discovered all these spots are mutations. So there's a background. That's the point. The cancer genome is full of mutations, and sorting out which ones are passenger and which ones are driver oncogenic is a challenge, but you'll find lots of mutations when you start looking. So BRAF is just a hotspot oncogenic mutation that we know plays a role, but that's the point of this project, is to find out what else is out there besides these known BRAF. So we also look for gene fusions. Remember, RET back a long time ago was discovered to come together and form a fusion. Well, in fact, we found 4 more partners of RET. So RET is rather promiscuous gene. Whatever RET fuses with can form an oncogene. As long as this part of the RET gene, which has the kinase shown here in green, is preserved and expressed, this will function as an oncogene. We discovered BRAF fusions, which is a big part of our paper. BRAF, again, is promiscuous. It likes that V600E hotspot, but there's other ways to activate BRAF and turn it into an oncogene. And you can see here's various genes. We found 9 different forms of BRAF fusions. We found ALK fusions. Those of you who know from the lung field, ALK is an oncogene or a gene in lung cancer that fuses with EML4 and forms an oncogene. And there's a drug called crizotinib that will block ALK fusion-positive lung cancer. So this has immediate implications. Now, we only found 4 of these, so it's about 1%. But if you're that 1% of patient who has advanced disease and has an ALK fusion, you will get treated with crizotinib. So that's a big, and then NTRK with ETV6, we found 5 or 6 of those. So fusions were a big part of our story. And notice, no cancer has more than one fusion. So they're mutually exclusive. Again, there's no point in getting more than one of these, right? And then when you put them together with the point mutations, the fusions and the point mutations are mutually exclusive. So it's telling us, again, biologically, you either get a fusion or you get a point mutation, but there's no point in getting more than one. And then we looked at copy number changes. And most of the genome of papillary thyroid cancer is pretty quiet. There's not really no changes. But we have a few groups that have loss of part of chromosome 22. We have a group that has a lot of changes, and then we have a group that has some changes. And when you put those together, well, before I get there, and the follicular variant has an enrichment of these copy number changes, okay, as shown in the red and the blue. So that's kind of interesting. And when you display these copy number changes in the same format, you can see here are the quiet ones, here's some, here's 22q cross. But it's more interesting when you put it all together, because now we have the point mutations, the fusions, and then when these end, you can see right here there's an enrichment of chromosomal changes. And we speculate, we did not prove, we speculate in our paper that these actually represent oncogenic driving events. Now, this is a half a chromosome. It's either gained or lost. There's hundreds of genes in each half a chromosome. Knowing which one is responsible is a project, unto itself. So we just went up as far as saying we think in some small percentage of cases, arm level changes can be drivers. Even when you look at the remaining few tumors that are still quote unquote dark, so Gaddy's a physicist, they like to talk about dark matter, right? So they talk about the dark matter of the cancer genome, which is basically tumors where you can't find a driving mutation. So we talk about the dark matter. And if you look at the dark matter, they have mutations. And some of them are known ATM and other cancer mutations. So when you get right down to it, we only had 5 tumors out of 402 that we had all this data on that we could not find a driving mutation. So we considered that a huge success, which will have implications for this whole field of molecular testing, because we're filling out the somatic mutational landscape of PTC. We went on to, yes?
Isn't one of the biggest advantages of this is alternative treatments when radioactive iodine doesn't
Yes,
no longer work?
yes, exactly.
If you find it has the same mutation as a certain lung cancer, you can use that.
Yes, exactly. So if you're lucky enough to have that mutation, right, that's the point of personalized medicine, is matching up your mutation with the targeted therapy. And so if you have the mutation, then yes. If you don't have the mutation, then those therapies don't work. Related to that point, though, is we showed that these mutations are clonal. What does that mean? It means that they're in all the cells. And why did we bother? Because there have been papers that suggested that BRAF is not in all the tumor cells, and therefore treating it with a drug that's going to block the activated BRAF is a waste of time because it's only in half the cells. But we showed, through a very sophisticated program called Absolute, that almost all the tumor cells have this mutation. So that's a big point that has implications for targeted therapy, and all these mutations are clonal. Yes?
Question about BRAF, and I understand that there's some common treatments among BRAF cancers, whether they're colorectal or
Yep.
melanoma. Those are kind of common things, and the treatments, the research in, say, melanoma designed to cover it.
Yes.
Now, I have a question sort of on the opposite end of that, which is, so if in your discovery of these oncogenetic phenomena, is it also true that somebody who has a BRAF thyroid tumor might also have general susceptibility to other BRAF cancers, or is that only on the treatment side?
So you have to separate somatic versus germline, right? And so BRAF is not very much a germline tumor. So that mutation occurs in your thyroid cell, and so it has no implications for whether it occurred in your colon cells.
So it doesn't, I'm just curious, like, sort of for screening or whatever, like if you have this BRAF mutation, so you're saying it's just arbitrary, like your germline cells aren't more susceptible to getting BRAF?
With the caveat, we could have in the hallway, the caveat. So if you have Lynch syndrome, right, or some ultra mutator phenotype where you're more likely to accumulate mutations, you're more likely to get a BRAF hotspot mutation, right? See how that follows? So they all tie together, but just because you have a BRAF mutation in your thyroid doesn't mean you're more likely to get melanoma or colon cancer, because those cells have to acquire the mutation by themselves because they're somatic, not germline. So this is a big statement for our paper. And then the other thing that happened in 2013, these TERT promoter mutations were discovered in various tumors, and it was a way to reactivate telomerase. And very quickly it became interesting for thyroid, and there's already several papers published that shows that the combination of a mutation plus a TERT mutation leads to a much higher risk of recurrence. So this will start showing up quickly in molecular diagnostics lab, and as a consequence, we really pushed the Broad to do the TERT promoter mutations. It was technically hard. I'm not sure exactly why, but they did it, and they did it in full force. They sequenced the TERT promoter mutation, TERT promoter 100,000 times per tumor. It was like incredible what they did. And we basically showed that in our cohort that TERT mutated cases are more likely to be high risk, that they have higher MESA scores, and they're less differentiated by a measure that I did when that we developed, which I'll tell you about. So together, TERT promoter mutations are associated with a high risk of recurrence, poor survival prediction, and low thyroid differentiation. So our study supports what's already been published, and I really do think that this will be in your molecular diagnostics lab soon. So this is a summary of the somatic alterations. I do want to get us out of here by 12 o'clock. Sorry for taking so long. So basically, we have a low mutational burden. Mutation density has some prognostic significance. We have a handful of driver mutations, some that are well known, BRAF and RAS, but we discovered a new one. We discovered diverse gene fusions. We showed that arm level, like half a chromosome, copy number alterations were present, and especially in follicular variant tumors. We suggested that these may be driving events, and this has implications for molecular diagnostics because these may not be so easy to figure out. We have a very small number of cases without driving mutations. We showed that they're clonal, and we showed that the TERT promoter was aggressive. Clearly enough for a paper, but we faced a challenge in the analysis working group. And again, I told you we just had to do papillary. As you know, papillary, compared to some of the other thyroid cancers, is relatively indolent. And to do the outcome really well, you need 20 years of follow-up. And our cohort was relatively newer, with just 2 or 3 or 4 years of follow-up. So it's not like we were going to do that 20-year, who've recurred in 20 years type of analysis. And this low mutation density challenged us. What other stories could we tell? So we sort of decided that we were going to sort of push the envelope. Some people said, let's just write up what we have, do a few more things, and be done. And most of us said, no, we want to keep going. So we defined the somatic genetic landscape of PTC, and then we leveraged all this to tell a more clinically relevant story. And we took advantage of the fact that BRAF and RAS were mutually exclusive, that the PTC genome was relatively quiet. We had all this data from RNA sequencing, microRNA sequencing, protein analysis, DNA methylation, and we had an incredibly brilliant group of people, computational biologists, who wanted to push the envelope. So we did. So again, BRAF and RAS don't occur together. So Giovanni Ciriello at Memorial and Katie Hoadley at UNC said, we can do something with this. And what they did is they developed a score that measures whether a tumor was more BRAF-like or more RAS-like. And they distilled it out. They pulled out 71 genes that allowed them to define this 71 gene signature. And then Giovanni did some fancy math and distilled this 71 gene signature into a score that went from -1 to 1, with negative numbers being more BRAF-like and positive numbers being more RAS-like. And he made this beautiful figure with the BRAS score, which we called the BRS, the BRAF V600E RAS score, BRS, and displayed the BRS in this blue-red continuum. And then he took all the mutations that we found and put them right underneath in this context, and showed that all the BRAF mutations, all the V600 mutations, are BRAF-like, as expected. That's how he designed the assay. and all the RAS mutations were RAS-like. There's one here that's cheating, but it actually has both, it has BRAF, EIF1AX, and RAS. So it's one of those triple mutants that breaks the rules, okay? But it's weakly BRAF-like. And then what's really cool about this is that we found these weird BRAF mutations. We found a few indels and a K601. They're RAS-like. So it's telling us that those mutations are creating a different signaling, a different phenotype. And then you can see all the other mutations we found, and you can see where their biology is. Notice all the ones that are strongly BRAF-like are V600E. There's no other weird mutations down here. So this told a beautiful story. And then when we brought in all the data from all the other platforms, put it together, you can see these RAS-like tumors are red consistently throughout, whereas the BRAF tumors are blue consistently across all these other genomic platforms. And this is shouting to us that these tumors, whether they're driven by RAS or RAS-like, or driven by BRAF V600E or BRAF-like, are basically different, have different biologies. So one of our conclusions of our paper is that we should break these up. These should no longer really be under the umbrella of papillary thyroid cancer. We then looked at thyroid differentiation. It was known that when you introduce a BRAF mutation, you actually get silencing of the iodine metabolism machinery. So just BRAF alone leads to a less differentiated state. So we got interested in thyroid differentiation. We found the genes that were involved in thyroid metabolism and developed another score called the thyroid differentiation score. And these are the genes that are primarily variable in that score: thyroglobulin, thyroid peroxidase, the symporter, some of the other genes. We have normal samples and the PTC samples. And we could see a big range in the cancers from highly differentiated, almost like normal thyroid, to loss of iodine metabolism machinery, less differentiated. Here's the TDS, here's the BRS. We also developed the ERK score, which I'll probably not mention, the histology, risk, and MESIS, and then the mutations. And we put those together, and now you can see, here's the BRAF V600E cohort, and you can immediately see in this box is there's a range of differentiation within the BRAF V600E cohort. This is really significant because the field up until this point has been considering BRAF V600E to be a uniform or homogeneous group of tumors. And this analysis shows, in fact, there's a lot of biological diversity in that cohort. Most are less differentiated, but there's a group right here that are more differentiated, more like the RAS-driven tumors and more like normal thyroid. So we have a broad range of differentiation. And so that just clearly suggests that there's biological diversity in this group. And so keep that in mind because we're going to get to the more formal molecular classification. We asked which genes are correlated to this score because it's pure math. We have thousands of genes and we could do correlations. And so we found a few genes that were correlated. KIT, and I don't have time to go through all of these, but I just point out miR21 and miR146, which are microRNAs that regulate the expression of other genes. They're small RNA molecules that regulate expression. And so we found some that were correlated and some that are anti-correlated. So keep those in mind. We moved then to molecular classification. We were able to show that the RAS-like tumors form a single group by their gene expression, but then we were able to find 4 classes of BRAF V600E tumors based on their different gene expression profiles. And you can see here's all the data. There's pretty good agreement on the RAS-like, and there's pretty good agreement over here. And so there's a robust signature that papillary thyroid cancer has different forms, different molecular subforms. We use this thing called supercluster, which takes all the data and spits out a single result, and showed in a hierarchical clustering the RAS-like tumors are very different in all their analytical platforms and from the BRAF-like tumors, and that you can start to see that there's a group here that are really looking like they have an agreement across protein, messenger RNA, and DNA methylation. And then we looked at the mirrors in detail, and this is the last figure in the paper. So we took all these scores that we developed, the BRS and the TDS. We took the histologic type. We took the tumor grade. I graded all the tumors. We took, and then we did the clustering based on their messenger RNA expression profiles. So we have a RAS-like group, and the RAS-like group are mostly follicular variant tumors, which is shown here, follicular variant, and they have high BRS values. They're mostly RAS-like, and they have high TDS values, right? So that's telling us that these follicular tumors that are driven mostly by RAS are really different across all their profiles, and it sort of reinforces what I just showed you. Then when we come to the BRAF side, we have actually 5 clusters here. One's really small. So now we start to see some of the genes, the microRNAs whose expression is driving this. And I have an arrow here at near 21. And I showed you a couple slides ago that near 21 was one of the nears that was highly correlated to the TDS. But resurfaces in this analysis and telling us that cluster 6 is really driven by near 21. Likewise, we have 146B, the other near that I pointed out, is driving another cluster. And to try to prove to you guys and the reviewers of the paper that these were biologically meaningful, we show that this group, near 21 group, has the lowest BRS scores of least differentiated tumors, just like 146, and that these other clusters have a mixture of BRS and TDS values. So the other point is this cluster has 3/4 of the tall cell variants, which are a known aggressive form of papillary thyroid cancer. So we think we have a great hypothesis that near 21 is one of the secondary drivers of the tall cell phenotype. We also show, I don't have time to show all the data, we also show that it's epigenetic regulated, means that the methylation affects its expression. So it might be that, your lifestyle impacts that in some way. That's really out there, but the data is the data. So we have this tall cell variant in near 21, and that's how we ended the story, suggesting that on top of these oncogenic drivers, that the expression of mirrors are fine-tuning the phenotype of PTC. And so just to recap what we did, we started with data from multiple genomic platforms. We defined the somatic landscape. We brought in the RNA expression data and the protein data to do this signaling and differentiation stories. We developed the BRS and TDS. We then turned to unsupervised clustering for molecular classification. We used the BRS and TDS measures along with clinical pathologic data to develop a more refined and we think is a more meaningful classification of thyroid cancer, these RAS-like versus BRAF-like, and then within the BRAF group, at least four groups of BRAF-like PTC. Cell, we're publishing this in Cell. They make you make a graphical abstract, which I made a couple of weeks ago, and this is what we put together. It's sort of the same thing, 496 cancers. We did all this data. We developed these scores using different parts of the data. We brought in all this to bear on the molecular classification, and now we're arguing that RAS-like PTC is fundamentally different from BRAF-driven PTC, and then we went on to show that there's different subgroups of BRAF PTC. So, major conclusions, there's really something for everyone in this, whether you like new mutations or signaling. We have new mutations. We showed that these different types are fundamentally different. We identified different subgroups, and it explains why BRAF V600E can't be the whole story. It's too common, but now there's too much genetic complexity in that group, and it's no longer appropriate to consider BRAF PTC as a homogeneous group, which there's 1,000 papers out there, but they're all going to have to be looked at again to capture this. And so our paper actually comes out and says a reclassification is justified. We don't propose the final solution. There'll be a working group of pathologists that can deal with this, but we didn't want to go too far. Clinical significance, again, we're catalyzing a discussion of the appropriate classification of in particular, the follicular variant of PTC. Yuri's organized this working group for the reclassification of encapsulated follicular variant papillary carcinoma because, in truth, a lot of patients with a small marble-like 1 centimeter nodule that I diagnose as encapsulated follicular variant of papillary carcinoma, at Michigan, my colleagues know, I'm shouting to them, go easy on this person. But that person still may get a completion thyroidectomy and in the right hands or the wrong hands get radioactive iodine, which is complete overtreatment, in my opinion. If I had a RAS nodule that was 1 centimeter and completely out, I would say, thanks very much, I'm done, goodbye. but that,
Are you saying that RAS is less aggressive?
so when in the right setting. So clearly RAS mutations are present in more aggressive tumors, but in the setting of it being diagnosed as papillary carcinoma, especially when they're encapsulated and non-invasive, those are more indolent. Yes, that's one of the statements. So our study clearly has an impact for, I'm almost out of time, I think, impact for molecular diagnostics. It already has impacted molecular diagnostics. We actually have, with Hopkins, Michigan, Cornell, and Mayo, a validation study underway looking at MIR21, 146, and a few others to predict lymph node positivity, to sort of just help surgeons decide whether people need central compartment lymph node dissections. So we're looking at prognostic avenues. Clearly, I think that there's enough data on TERT that will be used. And then I think on impact on clinical trials going forward, it's no longer going to be adequate to enroll patients with papillary thyroid cancer. You're going to have to know what is the underlying genetic basis of that papillary thyroid cancer, because there was one study with a MEK inhibitor published in the New England Journal by Dr. Fagan that showed that patients that had RAS-driven tumors with the follicular variant responded preferentially than tumors with BRAF. So, for radioactive iodine, it may not be crucial, but as we enter the realm of targeted medicine with targeted therapy, precision medicine, then knowing this information will be needed. So here we are, again, at the same meeting, but now with the paper published in Cell. It was not easy. It was initially rejected because in the last minute I'll tell you, so basic scientists fundamentally hate this stuff. And one of the persons who reviewed the paper, I could read between the lines. He emotionally said, "I hate this," and said, "There's no science, there's no biology here. Why?" He said, "Why would you even make classes of PTC if everybody is cured 95% with radioiodine? It's silly." And so it was such a bad review, though. It gave us the leverage because the other review was quite good, and it was rejected on the basis of one review. And science is if you get one bad review, you're usually done. But this is the TCGA. So we wrote a letter. The editor agreed to talk to us. This was back in June at the Endocrine Society. I went through the whole paper, explained the significance of the paper. We had the head of the TCGA call in. Gaddy went ballistic because one reviewer said, "Why did you bother with 500 tumors?" And Gaddy's like, "Every other paper I've ever done said, you didn't do enough tumors, do more." And finally someone's saying, "Why'd you do 500? So after a couple of days, the editor agreed to send it out for 2 more reviews, and they both came back glowingly, and she changed her mind. So we were very excited about that because it's a massive paper. It's almost 7,000 words. The other place to put these is Nature, but they like 3,000 words. And so the hard part is all these people do this analysis, and then as the co-chair, you have to cut and say, you did a great job, but we have to take you out. And I guess I'm a softie, and we found a way to keep everything in. We even had to put some things on the TCGA website. We had to put a few supplemental figures, but there's 7 figures. There's 10 supplemental figures, each multi-paneled. So there's about 75 figures in this paper if you include even the things we put on the supplemental website. There's probably about 25 tables. So it's a massive thing. Question?
I just want to make sure I understand it. The 2 types, you think that papillary should go into 2 types, those that have the presence of BRAF and those that have the presence of BRAF. But they both can be treated with radioactive.
Yes, yeah, oh, absolutely, yeah. Well, exactly.
But if they are persistent and recur and are not differentiated,
right.
then the drug that would be recommended to treat them would be more targeted therapy because
That's the general direction that we're headed, right, as we discover these. And so the big factor is drug development is still a 10-year process. And drug companies have been working on these. So there are a variety of MEK inhibitors that block the BRAF pathway coming out. They're being treated for so the big field is melanoma. But things that happen in melanoma, like someone pointed out, are immediately at least applicable to the thyroid because melanoma, colon cancer, and thyroid have the combination of BRAF and RAS mutations. Colon's a little more complicated, but and so that's the direction we're headed for people with more advanced disease. So this paper has messages for all levels, for people with nodules, people with more advanced cancers, and this will be the foundation So we didn't actually convince the TCGA to do aggressive thyroid cancer. We went back and forth, and the question was, is there someone out there that has a freezer full of 100 anaplastic carcinomas? I have 5. MD Anderson has, probably a few more, but we just could never get it off the ground. But that project's yet to be done. But this will be the foundation upon which everything else is compared. And in fact, I know there's a study being done at the NCI, 400 tumors of papillary thyroid cancer from the Chernobyl, Ukraine area, and that will be directly compared to this, and they've asked us. I think the Broad is going to do the sequencing. So here's the paper. We're really excited that it's finally coming out. And again, Cell allowed us to tell the full story. So it's really 3 papers all woven together. This is really the work of a lot of people. I know Gaddy and I get a lot of the credit as being the co-chairs, but this person right here, Chip Stewart, is in Gaddy's group. He's the analysis coordinator. I can't tell you the emails that I got at 2 in the morning from Chip saying, I just finished this, I just finished that. I mean, the guy was just amazing in the commitment. So a lot of people, 239 authors, I think, on our paper last time I counted. And you can recognize some of the names that enjoyed the ride here that were disease experts on the analysis working group. So I'll be glad. I know I've used up all our time. It's 12:01. I'll be glad to take more questions now, or we could do it out in the hallway.
Yeah, I think we're going to have,
I know it's lunchtime, and I know there's another session that starts at, 12:15? Yep. There's a question there. But thank you for your attention.
thank you so much.
Hopefully it was understandable.